Thank you for inviting me to talk. Today, I'm going to talk to you about the genetics and genomics of kidney diseases. I come from the background of human genetics. I'm a nephrologist adopted into urology. And so I'm going to talk to you about how we've been using the sequence of the human genome to be able to make discoveries and how we have opportunities in benign urology. The first draft of the human genome was published about 20 years ago. It took 10 years, $3 billion to produce that first draft. And since that time, with the advent of new sequencing technologies, we're able to sequence individual genomes for a few $100s, such that now hundreds of thousands or millions of people actually in many ways have had some aspect of genome sequencing or profiling done. And really the real challenge that we have now is to identify those rare typos in the genome, which then result in clinical phenotypes or traits that we are interested in. And so my talk today is going to talk about how we've been using some of these to get an insight into many complex disorders, but there are many, many opportunities yet to be explored in benign urology. And so the paradigm that's been explored for the most part in human genetics is the following. On the left, you have the paradigm of the monogenic disorder, where there's a rare variant with a large effect, which has a deterministic effect on the development of disease. So these are your typical monogenic disorders, such as polycystic kidney disease or cystic fibrosis, single variant with a large effect, which then has confers a very large probability that an individual carrying those variants will wind up developing a disease. On the other side of the spectrum, you have the polygenic, more complex determination, where you have the aggregation of common variants, which each impart a small effect on the risk of the disease, but which together tip the balance towards disease. And so now with the advent of genome sequencing, one can comprehensively explore those both paradigms and also look at the interaction between common and rare genetic variants. So in benign urology, I would say that so the monogenic disorders have really been mostly explored in the context of rare developmental disorders of the kidney, so essentially in pediatric urology. And in fact, there are more than 300 genes of genomic disorders that have been described in the literature that result in some form of congenital malformations that cause also some form of urologic disorder. And so there are many such genes that have been identified and cataloged and been published. And there are also genomic disorders, in other words, deletions and duplications of the genome that also result in multiple organ malformations, including genitourinary malformations. Today, earlier in one of the poster sessions, you heard from Greg Whitmore from our group, who described the 16p11 microdeletion syndrome, which results in multiple malformations across the body, but has a significant impact on the lower urinary tract and is associated with multiple GU abnormalities. And we believe that the deletion of one copy of this gene called TBX6 is really critical for the development of those abnormalities. On the other side of the spectrum, when we look at common variants, which have a small effect, those have not been explored really significantly in benign urology. Two good examples of those, however, are for hypospadias, in which GWASs have been done. And it's interesting that there are multiple variants that have been identified in genes that have been implicated in the urologic development, such as EYA1 or the Hox genes. But also there's a gene called DGKK, which imparts a fairly large effect on the risk of disease, even though it's fairly common. So this defies the common variant in a small effect paradigm. Similarly, we recently explored the contribution of common variant to vesico-ureteric reflux, and we identified common variants in developmental genes that are associated with risk of VOR. But there's also variants in one gene, WDPCP, which is involved in planar cell polarity, which imparts an odds ratio of about 3 and has a significant impact essentially on the development of vesico-ureteric reflux. But across the range of the developmental disorders, I would say that most of the phenotypes have not been explored by GWAS, and particularly for adult onset disorders that have been described today in this conference so far, most of the time sequencing studies or GWAS studies have not been performed, or if they've been performed, the sample sizes have not been adequately powered to be able to get an answer. So how can we try to address some of these? It's by perhaps using biobanks. The traditional model consists of recruiting a large number of individuals with a disease of interest, let's say MBPH or CAKUT or interstitial cystitis, and then getting a set of controls and doing a case control study. On the other hand, if you can try to exploit now biobanks, which are disease diagnostics and are essentially a collection of biosamples and clinical data, mostly electronic health records that have been collected across the health system or multiple health systems. And as a result of this, now you can select individuals with a suitable diagnosis, and then select individuals also for the biobank that do not have those diagnoses that can serve as controls. And you can then conduct multiple studies in parallel, for example, for CAKUT, for BPH, or for IC, and explore essentially the genetic contribution for these data if these samples have also been sequenced. So another way you can do this is, so you can do GWAS, that's what I described just now, but you can also, once you find a genetic variant of interest, which is associated with a disease, as I'm showing here, then you can turn this on its head and see, and because you have access to electronic health records, you can scan the phenome, essentially all the electronic health record codes, the diagnostic codes, and do a phenome-wide association study. In other words, now you can look at this variant and see how many different clinical phenotypes this is associated with, and get a sense of pleiotropy. And essentially you can do a GWAS by PheWAS across the genome and across all the pheocodes to be able to get an insight into this. So let me give you an example of biobanks that are out there that we can use. The UK Biobank is one of them that's not publicly available. There are tons and tons of papers that have been published, but very few actually that have been taking advantage of the benign urology traits that are documented in the UK Biobank. These are 500,000 individuals that have had GWAS data, and about 200,000 of them have had exome data publicly released. eMERGE is another network that I contributed to and we worked on in the past, where we have 100,000 individuals with genomic data and EHR data. The Biobank Japan. Another project that I'm involved in is the All of Us program, where we're recruiting over 1 million individuals in the United States for the precision medicine research. And so far, 300,000 individuals have been recruited. There are about 100,000 individuals whose genome have been sequenced, and this data is going to be made available to the public for essentially doing this type of research. Other ones include the Pakistani genome research, which is a rare, essentially underserved population, with 300,000 individuals that have been recruited to date. So what can you do with some of this electronic health record data to be able to sort of hone in on some of these genetic disorders? Well, you can try to combine some of the clinical traits that have been documented in electronic health records and some of the codes to be able to see if they all fit together and fit the pattern of specific Mendelian diseases. And this is what was done, for example, in this study published in 2018 from Vanderbilt investigators, where they tried to come up with essentially a phenotype risk score for Mendelian disorders. And as a result of this, we're able to identify a large number of cases of unidentified Mendelian disorders, such as hemochromatosis or Marfan syndrome, based on their phenotypes. And once they genotyped them and sequenced them, they realized that indeed they did have this genetic disease. So one can potentially do the same thing for urologic conditions that are poorly described or may not be picked up based on the usual clinical evaluation, because patients go to multiple doctors who may not be attuned to some of these urologic phenotypes that we're interested in. And by putting these electronic health record data together, one may come up with electronic phenotyping that might help identify patients a lot better. So, so I told you about the PheWAS approach. And so one example of this is one when we wind up putting together, my colleague, Christoph Kirlock, put together an electronic phenotype for CKD or chronic kidney disease, looking at a large number of data available from the EHR, and identified, looking at about 1 million people, and identified 200,000 people roughly who had chronic kidney disease. And incredibly, only about 20% of them had a diagnostic code for CKD. So in other words, there were a lot of people whose chronic kidney disease was missed, or at least not documented in electronic health records. And then because there was genetic data available in a subset of these individuals, he was able to do GWAS and picked up a replicated an association, a known association with uromodulin, which is the TAM horsefly protein, the most abundant protein in the urine. So what he next did, because we had access to electronic health record, he turned this variant on its head essentially and tried to then scan the phenome to see what other phenotypes this particular variant is associated with. And of course, recovered chronic kidney disease, but also identified acute cystitis and kidney stones as associated phenotype, highly associated phenotype. So these are novel phenotypic associations for this particular variant, demonstrating that there are multiple different discoveries that can be made this way by using the electronic health records and genome data. So I think that
Dr. Gavari, we have about 30 more seconds.
Okay, so we have a lot of opportunities in benign urology to be able to do this by collaborating right now with benign urology networks that are out there. On the right side of the screen, you see benign urology networks that the Columbia group is collaborating with to be able to advance genetic investigation of some of these disorders. And I would say there's just a lot of significant evidence for rare monogenic diseases among developmental urologic diseases. There are multiple opportunities for clinical implementation, but the contribution of common variants to these disorders has not been fully investigated. We have to really try to do a comprehensive genetic analysis for adult onset benign urologic condition, and collaborations with biobanks, the NIDDK networks, and patient networks will help in that regard. And being able to develop these electronic phenotypes for benign urologic conditions would also help. And we can then subsequently overlay this with omics data to get better insight into disease pathways. And I want to thank all the contributing networks and colleagues who helped with this project. Thank you.